Thus far in lecture, we've been talking about membrane proteins and the types that are possible within a lipid bilayer. However, what if you don't know what type of membrane protein you have? This animation is here to show you one type of technique we can use to determine if a membrane protein faces the extracellular side of the cytosol or the cytosolic side, and whether or not it can be a glycoprotein. With small variations to this protocol, we can additionally determine if it is a transmembrane protein or a monolayer associated protein, but these won't be explored in this animation. There are many different types of membrane proteins, and here are just a few of them. You'll notice that some of them have exposure to the extracellular side, and some don't. In addition, there are proteins that have sugar groups added. These are the things shown in blue. When we have these sugar groups added, these proteins are called glycoproteins, glyco meaning sugar. In this scenario, we have a cell and we have three different proteins that we're interested in determining their orientation. We're interested in determining if they're extracellular facing and if they have carbohydrate groups making them glycoproteins. So here's the experimental setup. We have three different cell cultures for the same types of cells that are grown. To each of these different cell cultures, a different compound is added. In one of the treatments, a compound called FM is added. When this FM reagent is added to the cells, any part of the protein with a portion on the extracellular side will have the FM attached, since it's only able to access the outside of the intact cells. In a separate set of cells, a compound called PAS was added. When the PAS reagent is added to the cells, it recognizes any protein that has a carbohydrate or sugar group on the outside of cells. For the last treatment, nothing is added. This serves as a control to make sure all our proteins are isolated properly in the following steps. Now that the cells were treated with the appropriate markers, the PAS and FM markers, the membrane proteins need to be isolated from the membrane environment. The way that this is done is by using detergents. When mixed with membranes, the hydrophobic ends of the detergents bind to the hydrophobic regions of the membrane proteins. This then displaces the lipid molecules. Since the other end of the detergent molecule is polar, the binding tends to bring the membrane proteins into solution as a detergent protein complex. The result of adding detergents is that the detergents replace the lipid bilayer and stabilize the hydrophobic membrane region of the protein. In addition, you get a byproduct of little lipid packets as well. In this scenario we're showing the proteins with no treatment. However, the cells that were labeled with FM or PAS would retain the label in solution. So the FM would remain attached to any piece of the protein that was originally on the outside of the cell. In the PAS treated cells, the PAS would still be bound to the carbohydrate units. Now that the proteins have been isolated from the membrane, the proteins in solution are heated and mixed with SDS. This heat in SDS causes the protein to linearize and is fully coated with a negative charge that is equal to the mass of the protein. The protein mixtures are put into wells in a polyacrylamide gel matrix. Then they are run in an electric field. Because they have been coated with a negative charge, they flow towards the positive terminal. The bigger proteins are slowed in the gel matrix compared to the small proteins, so they separate based on size. To make the gel easier to work with, the proteins are transferred to a special paper, which then can be developed to see the different proteins. In this case, we would cut the gel into 3 pieces and add a solution that recognizes the FM molecule, the PAS molecule, or binds to all proteins. The solutions were added to detect the markers and the bands are developed. Now that you understand the protocol, you should be able to analyze which of these proteins here have an extracellular component and which are also glycoproteins.